Hello, the question statement says, given below is a pedigree showing the inheritance of autosomal dominant disorder. And the pedigree is like this, and the options given are 1. There are 3 generations given, and the first generation shows capital M, 2, capital M, small m, 3, small m, small m. And the B option is, first is capital M, second is capital M, and third is small m, small m. The C option is, 1 is capital M, small m, 2 is capital M, and 3 is capital M, small m. And the D option is, 1 is capital M, small m, 2 is capital M, small m, and 3 is small m, small m. So this is a pedigree analysis. And what is pedigree analysis? It is the study of the inheritance of genes in a family. And here we have given 3 generations, and in all generations we can see the affected individuals, but not in the case of second generation. It is not the offspring affected, but it is from the outside. Only the mother is affected. So the offspring of the first generation is not affected. And here in this question itself it is given, it is an autosomal dominant disorder. If it is autosomal dominant disorder, what are the clues we need? Autosomal dominant clues are, it does not skip generations. It don't skip generations. And what is the other clue for autosomal dominant disorder? It is affected parents can have unaffected children. Affected parents can have unaffected children. So in any pedigree analysis, what we look at the first time is whether it is autosomal or sex-linked. Here all the sexes are affected, so it's definitely not sex-linked. And autosomal, we need to check whether it is dominant or recessive here, but in this case it is clearly given it is an autosomal dominant disorder. And what are the genotypes of these 3 given? That is 1, 2 and 3. Here in the question we need to give the genotype of these 3 people. To how to find the genotype? We know affected can have a trait of capital M, and capital M, small m, and the unaffected remains as small m, small m. So here in this case these can be capital M or capital M, small m. But here 3 out of 4 children of second person is affected. So definitely the second person should be capital M. And here this is clearly unaffected, and which remains small m, small m. So we got both these. 2 is capital M, 3 is small m, small m. What all option says this condition? 3 is small m. We can see that in these 3 options. And the second one is capital M. We can see in only this. And definitely, and the person here is unaffected. But the third person is clearly unaffected, and the second person has to be capital M, so that 3 out of the 4 children remains affected. And for this, if 3 out of 4 children should be affected, then definitely the father should be a carrier. If father should be a carrier, then definitely the person of the first generation, of the father in the first generation, has to be capital M. So the option is B. The correct option is option B. 1 is capital M, 2 is capital M, and 3 is small m, small m. Hope this answered your question. Thank you. Hello. The question statement says, which of the following 2 out of 4 statements are incorrect? And the statement A is, the first transgenic buffalo, row C, produced milk which was human alpha-lactalbumin. The statement B is, restriction enzymes are used in the isolation of DNA from other macromolecules. And statement C is, downstream processing is one of the steps of rDNA technology. And statement D is, disarmed pathogen vectors are used in the transfer of rDNA into the host. And the options given are, option A, statement A and C are incorrect. Option B, statement B and C are incorrect. Statement C, Option C, statement C and D are incorrect, and option D, statement A and B are incorrect. Now let's check which all statements are correct. So statement A, the first transgenic buffalo, Rossi, produced milk which was human alpha-lactalbumin. Yes, Rossi is the first transgenic buffalo in 1979. So Rossi is the first transgenic buffalo. That is correct. And the second part of the statement is, it is human alpha-lactalbumin. Yes, that is also correct because Rossi was cloned to produce human alpha-lactalbumin. So the statement A is correct. And the second option is, restriction enzymes are used in the isolation of DNA from other macromolecules. What are restriction enzymes? Restriction enzymes are nothing but proteins derived mainly from bacteria that are used to cleave DNAs which are not desirable. So the restriction enzymes acts as cutter enzymes in the DNA process. So here the statement says restriction enzymes are used in the isolation of DNA from other macromolecules. That is a big no, because restriction enzymes can be only used in the pure form, pure molecules only, not from the macromolecules. For the isolation of DNA from macromolecules, we need a pre-treatment. That is, cellulase are used to treat plant molecules, chitinase for fungus, lysozyme for bacteria, and proteinase K for mammals. So after that pre-treatment only, the DNA isolated is then treated with the restriction enzymes. So as the statement says, restriction enzymes are used in isolation of DNA from macromolecules, we need a pre-treatment. That is, cellulase are used to treat plant molecules, chitinase for fungus, lysozyme for bacteria, and proteinase K for mammals. So after that pre-treatment only, the DNA isolated is then treated with the restriction enzymes. So as the statement says, restriction enzymes are used in isolation of DNA from other macromolecules is a big no. So that option is wrong. Then comes the statement C, which says downstream processing is one of the steps of rDNA technology. First, we have to stick on to this steps of rDNA technology. What are the steps of rDNA technology? It is the cloning of desirable genes, its replication, like the selection after it's transcribed, the transcription and the translation of all those genes. Those comes in rDNA technology, that 4 to 7 steps. And downstreaming is not a step in the rDNA technology process. It is the purification of the synthetic, after the fermentation or the pharmaceutical manufacturing. After that, the purification is what is called as downstream processing, and that is not a step of rDNA technology. So that is also a big no. Then comes the statement D, which says disarmed pathogen vectors are used in the transfer of rDNA into the host. What are vectors? Vectors are carriers, vehicles which are used in the transfer of rDNA into the host. That is correct, and disarmed pathogens, we know clearly it is a vector used. That is the most popular vector of disarmed pathogens, like the plasmid TI and all. So disarmed pathogen vectors are used in the transfer of rDNA technology. Why is it disarmed? It is after the that pathogenicity is deleted, and that pathogen is used for the transfer of genome into the host, and that has also a wide applicability too. So that statement is correct, and from this we can see that option B and C are incorrect, which is said in the option B, that is statement B and C are incorrect. Hope this answered your question. Thank you. Hello, the question statement says, which of the following statement is not true for retroviruses? Option A, DNA is not present at any stage in the life cycle of retrovirus. Option B, retroviruses carry gene for RNA-dependent DNA polymerase. Option C, the genetic material in mature retroviruses is RNA. And option D, retroviruses are causative agents for certain kinds of cancer in man. So what we need to find is, out of these 4 options, which is not true, that is, which is a false statement. What is retrovirus? Retroviruses are viruses whose genetic material is RNA. Whose genetic material is RNA, but on reaching a host, what it do is, it just make a copy of DNA, just get inserted into the host, and make a DNA copy of its genetic material, and then it infects the host. And these retroviruses cause many diseases like HIV, AIDS, or the human T-cell leukemia like cancer, and many such diseases, and these are mostly deadly. So let's check the options. So here, which are the clear-cut correct answers? We can see that retrovirus carry gene for RNA dependent DNA polymerase. Yes, that is correct, because it makes from RNA, it makes DNA in the host genome. So for that, it requires RNA dependent DNA polymerase. So that is a correct answer. And retroviruses are causative agents for certain kinds of cancer in man. That is also correct, because the retrovirus cause diseases like human T-cell leukemia. So retrovirus cause cancer. That is also true. Then these two are the confusing statements. DNA is not present at any stage in the life cycle of retrovirus, and the option C, the genetic material in mature retrovirus is RNA. What is the genetic material in retrovirus in RNA? After maturation is like it enters into the host and it makes DNA. So this is wrong. What if we check DNA is not present at any stage in the life cycle of retrovirus? Life cycle of retrovirus, that is true, because DNA retrovirus DNA inside the host cell, and it is not the life cycle of retrovirus. So DNA is not present at any stage in the life cycle of retrovirus is also a correct statement. So option A, B and C are correct, whereas the option C is incorrect. So the answer is option C, the genetic material in mature retrovirus is RNA is not correct, and the genetic material in mature retrovirus is DNA. So hope this answered your query. Thank you.